historicity of the Shroud, a group of scholars and researchers traveled to the Renaissance Cathedral of St. John the Baptist in Turin, Italy, to examine and study the Shroud utilizing advanced scientific research techniques. Several of these experts participated in a symposium held at Elizabethtown College, Elizabethtown, Pennsylvania, where they presented their varying viewpoints on the issue of the Shroud as an object of archaeological significance. The symposium was jointly sponsored by the Cultural Events Committee of Elizabethtown College and the Marguerite Ayer Wilbur Foundation of Santa Barbara, California. It was organized by Dr. W. Wesley McDonald, Assistant Professor of Political Science at Elizabethtown College. This program is the first in the series of 7 presentations on the Shroud, videotaped at the Mystery of the Shroud of Turin Symposium, and features Dr. Eric Jumper from the Department of Aeronautics and Astronautics at the Air Force Institute of Technology, Wright-Patterson Air Force Base, who is noted for his research in the area of laser physics and gas dynamics.
Well, let me start by saying that I had planned to wear a white shirt, a tie, and a coat, but I came from the airplane to here after having slept in my clothes, and then on the airplane I had the fortune of having the stewardess dump a Coke on me. So we'll see what comes off. I had planned to spend last night just going over some notes that I've been making over the last week, but you'll just have to settle for what I've got here. Now, for a long number of years now, I've been involved in a controversial research project. The controversy isn't new. In fact, you can track papers back to the 1930s that have controversy in them. By now, there's all sorts of wild hypotheses on the cause of the phenomena under study. There's lots of demagoguery. There's lots of embedded positions. There's careers that have been built in this area. There's empires. There's lots of reputations at stake. Recently, we, and I say we by I and some of my co-workers, have produced an extremely discriminating set of data with enough detail to begin to sort through a number of these hypotheses. The data has been presented at a number of technical meetings and published in technical refereed journals and is now referenced extensively. Rational theories may now be tested for compatibility. Many theories have been rejected for lack of compatibility and what is now known about the phenomena. Other theories had to be modified by enough ad hoc caveats that they now seem quite improbable. Now, you probably think that I'm talking about the Shroud of Turin, but I am not. I'm talking about an area of study that I've been involved in called dynamic stall. Has nothing to do with the Shroud, but it has everything to do with a research scientist's career. Because what we do is we try to answer questions that have not been answered before. It's not an applied field. It is a field that attempts to answer unanswered questions. There are a lot of phenomena, like dynamic stall, that are repeatable in the laboratory, and people still don't know what caused them. So it is not unusual that something like the Shroud, which is a single thing, a single event, produce some kind of image like this, either by artist hand or whatever. There are no other things like it. We can't reproduce it in the laboratory. So it is not unusual that there would be controversy involved in this area. It poses a lot of unanswered questions. Now, the only big difference, I think, between what I've mentioned to you before and the Shroud, well, there are really two big differences. One of them is that the Shroud involves a religious connection because of the depiction of what appears, all of the injuries and so on the man depicted there are similar to those described in the Gospels for Jesus of Nazareth. So you have this added dimension of complications and new kinds of embedded positions on the topic because it has a religious nature. The other thing is that I have never been involved with a research project that has come under such, let me call it, media abuse. I've never read anything in the popular literature referring to the things that I've done that have been correct. And I've never really understood why my character has been attempted to be assassinated in the media. And I think probably some of this is my own fault because we were quite naive in the beginning, having dealt with other research projects but not having dealt with the press. So if that's the reason that I'm taking this kind of abuse, then I apologize for that. Maybe the abuse will stop with that apology. I doubt it. With regard to the Shroud and science, let me make it clear from the beginning that science is in absolutely no position to prove the authenticity of the Shroud. And if you came to hear that there's some kind of proof for its authenticity, then you've come to the wrong place, because it won't come from me, and I doubt if it'll come from the other people that have been working with it. And on the other hand, there are a number of people that say science has disproved the authenticity. This I do not believe. But I will say that science is in a fair position to disprove things. And in fact, that's the best that it does. But in the case of the Shroud, as I think will be made clear to you in a number of presentations today, I don't believe that the data is there to show that the Shroud is not authentic. It still could be. Personally, I don't know whether the Shroud is authentic or not. It seems too good to be true. I tend to believe it probably isn't authentic. But on the other hand, I recognize that there isn't anything that we've come up with or anybody else that eliminates that possibility. Now, in regard to the involvement of the Shroud of Turin Research Project, incidentally, I told you this is hasn't gelled exactly. I was hoping it would gel last night. But concerning the evolution of the Shroud of Turin Research Project, it basically evolved from a meeting in 1977, and it evolved from a number of people that were curious about the Shroud and had done some things that had something to do with science on it, and had actually raised a number of questions. And so in 1977, we felt there were enough people sitting around out there that were interested in this that, in fact, Father Otterbein, who's sitting out here, the president of the Holy Shroud Guild, sponsored a meeting in Albuquerque, New Mexico. And actually, it was quite a meeting. There was a large turnout. There were more people interested in the Shroud that had done a few things on the Shroud than I really realized. And we had a nice day presenting this preliminary type of work to one another. On the second day of that meeting, there was a workshop. And the workshop basically tried to take a number of the questions that were raised in these preliminary studies that all sorts of people from all over the world had done, and see if there is some way that science could address these questions. Of course, the natural thing that came out of this was that we decided that science could at least try to determine what the image was. At that time, there were all kinds of rumors about what it might be. And if you look at what came of that second day, you will see that what came out of that was a series of tests, a whole battery of tests, which I'll describe to you in a moment, to try to address the question of what is on the Shroud. What are the images made of? Is there a difference between the blood images, for example, and the body image, and all sorts of things that came up this way? And just like I mentioned with the dynamic stall problem, our feeling was that someone had to produce a good set of data against which theories for the cause of the image could be compared for compatibility. And that's what we tried to do. We tried to produce a discriminating set of data. Now, I find it somewhat hard to believe when I read in the media that what we were trying to do was to prove the shroud was authentic, or to prove some religious kind of belief that we had, because that just isn't what we tried to do. It isn't the history of what we've done. It doesn't speak to what we've done with the data. It doesn't have anything to do with the facts. It just has to do with the kind of abuse that the media has given this area. And it's not surprising that those people out there that are interested find themselves quite confused. I gave a talk in Kansas City to the American Chemical Society, national meeting on this topic, Al Adler and I. We were told by those in attendance that it was the best received paper of the entire meeting. It was the first time that they'd ever seen a paper given on the last day, at the end of the last day, that was absolutely jammed to capacity. It was the first time they had that many people stay to the end of the meeting. As I say, it was very well received, and I went to the airport feeling quite good about that, only to find a copy of Discovery magazine where our character was attempted to be assassinated. I don't understand it. I don't know what we've done. Would have been nice if the shroud turned out to be a painting. That's actually something we might have been able to tell. But it didn't turn out that way. That's the way it is. Let me just talk to you a little bit about what it was that we did do. Thought I had some more notes here, but I can't find them. But I do have some slides, and I'll talk from those slides for a few minutes about what it was we decided to do in 1977 in the way of data collection. And then following that, I'll just sort of briefly overview what we found. And I won't go into much detail because you'll hear a lot about this from Al Adler that will cover the chemistry in great detail. So I'll just sort of overview the findings, and then I'll talk for a minute about what can be said from those things and what can't be. So if I can have somebody turn that on. Well, first of all, that meeting, as I said, in March led to a proposal, and to our, I would say, utter surprise, the proposal was accepted. A lot of credit for that goes to the Holy Shroud Guild and to Father Otterbein and Father Rinaldi, who are the president and vice president of the Guild, for acting as intermediaries and getting us in to talk to the right people and so on. But let's talk for a minute about what it was we did in 1978 based on what we had decided to do in March of '77. Well, first of all, we did direct examination of the shroud. And I want to emphasize that, because while some other people had looked at it for a few hours in '69 and '73, we had really prepared to go in and do direct examination. And when I say we had really prepared, we had an operating plan that planned out minute by minute every moment we'd have with that shroud. We were not in the presence of the shroud trying to decide what to do. We knew we had a limited amount of time. We knew we were making direct examination. We're not likely to do it again. And so we tried to make the best use of our time. But remember, we were also trying to cover as large a ground as we could, to produce as large a data set as we could, because we didn't know what was on the shroud. Now, in regard to what we did, first of all, we did a number of photographic studies of the shroud. We photographed the shroud with some color film, mostly with black and white film through a number of filters, so that we held the camera stationary, and then we were able to take black and white photographs through some fairly narrow band filters. The idea being that we could get a crude spectrograph of the shroud, that is, a crude fingerprinting of the different wavelengths that we had collected on the film. This is rather important because it allowed us to generalize a number of the findings from specific tests. This black and white, this photography also included some fluorescence work, both UV and IR. I'm going to go back now to that because that just indicates that we were exposing the shroud to one color of light and collecting the information on the film through a different filter. So instead of seeing the reflection, we were seeing the emissions that were stimulated by the direct illumination from a different color. This is kind of like when you go into a sign shop and you see different colors under what they call black light or near UV. We also did some transmission photographs, that is, illuminating the shroud from the back and taking the picture from the front. I'll refer to those later. Don't have a picture in here. We also did some X-ray transmission. Again, this is over the entire shroud. That is, we use soft X-rays, which are not too unlike the kind of X-rays that you would get if you broke a bone, but they are lower energy, take longer exposures so that we can get fairly good resolution. We did that over the entire shroud. We also did some selected points where we did a very good job at visible spectroscopy, again fingerprinting, but now over very narrow wave bands and over the entire visible spectrum. We also went into the UV. We also took some fluorescence spectra. We also took some near IR, and in addition to that, we took some further out IR. We did also, again, and I want to make this point, we did direct microscopic examination of the shroud itself. In other words, we had microscopes there. We could go up to the shroud. We could probe the image area with dissecting needles. We could see what the consistency of the fibers were. We could see whether they were cemented together and that sort of thing. We did that directly on the shroud, not on samples that we took, but we did take samples as well. I'll mention that in a moment. Occasionally, we would take photomicrographs, that is, photographs through the microscope, for the purpose of documenting a characteristic that we had noted in our lab books that we brought with us, and which were characteristic of some of the things that we saw through the direct examination through the microscope. In terms of fluorescence, as I mentioned to you before, putting one wavelength on, collecting information at other wavelengths. We also did X-ray fluorescence, again at specific locations, and this is why I say that it was important that we had the sort of crude spectroscopy of the overall photograph so that we could correlate what we find there to similar locations on the shroud. In addition, we took some samples with sticky tape by using that device that you see in that gentleman's right hand, placing tape on the shroud in specific locations, then carefully removing those tapes and putting them carefully onto a frame and sealing them then in a hermetically sealed box and bringing those back for later analysis under the microscope using all sorts of electric and wet chemistry as well as visual observation. And I want to make the point here that the microscopic samples that we took are but one of an entire battery of tests that we ran, and the results of those must be embedded in the results of the entire set of data that we took. Now, in terms of what we found, well, we found all kinds of things on the shroud. I don't recall myself seeing a sample that didn't at least have one rodent hair. Note that no one, to my knowledge, has said yet that the shroud image was made of rodent hair, but I expect it any day. I want to talk about two kinds of images that are of most interest. There's really three that I'll mention. Ones of obvious most interest are the blood wounds. I don't have anything to sort of point with here, but probably everyone that is interested in the shroud enough to come here knows about those things. But we have blood images. We have a body image on which these blood images are superimposed. And then the last thing that I'll mention are some water stains. There are also all kinds of burn marks, patches, and other things, but I'll not address the results of those. But in general, we found a number of things. First of all, there was a lot of misinformation around before we went over there, and I find that that misinformation is still quoted and used as evidence for one thing or another. But one of the pieces of misinformation was that the blood was only superficial. That is, it didn't soak through the cloth. Well, this picture of a mirror looking up on the back of the cloth shows you that those blood images soak through the cloth, and this is quite different than the body images you'll see later. But it did soak through. This is one of those transmission photographs. If you look at the images, this happens to be the blood image on the wrist. They look like blood, so the medical people tell me. And notice that this also demonstrates that a good deal of this blood-looking material resides inside the cloth. That is, it's between the fibers, soaked into the fibers, goes all the way through the cloth. And in fact, it's much darker in transmission than it appears in reflection. So there's a lot more material, quite a bit of material in the cloth there. If you look at I don't remember exactly what this is, about 20x, so it's magnified some more. I really don't know what magnification you're looking at it. But this is what a blood image looks like. You can see that, in fact, most of the material resides down in the fiber, and you see a lot of the material is gone from the surface. This is kind of important because, as I'll mention in a few moments, that blood material had to go somewhere, and it's been chipped and bladed away over the years by folding and unfolding and other such things. And it's probably migrated to all over the cloth, and indeed we've found material like the stuff in the blood area literally all over the cloth, including in the control areas off the image. A little closer magnification, again you can see what I'm talking about. But also when we probed these fibers with a dissecting needle under the microscope, it was quite clear that they were matted together, cemented together with something. The fibers were coated. And again, you can see just how much material is actually gone from the surface. Again, most of it residing, embedding under the weave pattern, etc. You look at the sticky tape, we get all sorts of particulates of a number of different colors. You'll hear more about these. Just go through a couple of things. You saw before some things that look kind of red. We've got all sorts of shades of reddish, brownish, even some kind of olive-colored particles in those areas. And interestingly enough, sometimes we get what we call shards, things that look a lot like a linen fiber, but in fact there's no linen fiber there. This is some of that coating that popped off when the tape was pulled, broke free of the fibers, and we find a number of things that look like this on the shroud. This is in an area near where the blood intersected with the scorch, and you notice how the stuff turns black. That's kind of interesting. Now, we've done chemistry on this, and I show you this beautiful man here. He's going to talk to you later. He's going back there, says, look at that beautiful face. Al has really speared up the chemical analysis of a lot of these things, and basically what we find there is blood derivatives, blood serum, blood porphyrins, all kinds of things that tell us that what we're seeing there is old blood. And you'll hear a lot more about that. It's not paint, it's not touched up, it's old blood. That's what it tests. Now, it even gives some indication that it's human blood, although not as positive identification as blood itself. Again, you'll hear more about this in the chemistry talk. Now let's go back to this image and let's talk about things that are not blood, and that is the body image itself. This is a much more difficult problem, as it turned out, although now that we know the answer, it's not so tough. But of course, we're looking for something to explain why we see an image, so you'd sort of expect that there's something added to the cloth that produces the image. First of all, this is a very bad picture in terms of color, but I'll show you others, and one of the things is that the body image is a different color than the blood image. It doesn't show up that way sometimes in color photographs because that only selects a couple of bands of RGB, what's that, red, green, blue color separation, and it sort of misses the differences between those colors, although some of these you'll see definitely. But we know we've taken spectra of these things, and there's quite a dramatic difference between the color of the blood image and the color of the body image. Now, I've shown you blood image area. This is one of the darkest body image areas. When you probe this area with a dissecting needle under a microscope, as we did, you find that there's no evidence of cementing together of the particles, of the fibrils. In fact, there's nothing at this magnification that suggests there's any particles at all on the fibers. And notice the color of the shroud, the body image. It isn't red, is it? I have no idea why so many people labor under the impression or tried to peddle the impression that the body image is red. It isn't red. The body image isn't red. How can I say that differently so that you understand what I'm saying? Body image is yellow. Let's take a look at a little closer. There's a fiber. It's a body image fiber. You have to look pretty hard to see any particles there, don't you? Every once in a while we see some little red particles embedded in the medulla of the fiber, right inside the fiber. I was a little surprised when I went down to the store and bought myself a piece of linen just to make some controls and found the same particles. And you'll hear more about that, but linen is a kind of a grassy substance. The fibers are embedded in the grassy substance. And the way that the other cellulose is broken away from the fibers is to soak it and let it rot. And as you'll hear later, cellulose is great at selecting iron right out of the water. So yeah, there's a little bit of particles every once in a while. But notice there doesn't appear to be any particles on the fiber itself. And furthermore, if this thing were in really good focus like you could do on a microscope, you'll see that there doesn't appear to be any coating on that fiber. In fact, in the joints, you don't see any sort of meniscus evidence that there's any kind of coating. But what you do notice is that the fiber itself is a little roughened. That is in a body area, body-only image area. That is where there's body image, but there's no blood image. And there doesn't appear to be any water stain or anything contaminating the image. Now let's go to this next slide, and of course you'll recognize particles like I've shown you. In fact, you've seen this slide before. I said these are blood particles and so on. Now let me say another thing that I hear, which is misinformation. There is no significant number of particles in body-only image areas. There's no more particles in body-only image areas than there is in off-image control areas. And this is proof of that. This is taken from a tape sample that was completely off the images. This is a control sample. And we have found some of the largest pieces of blood material sometimes in some of these control areas. Now, I've just shown you a body image area with no particles, with an image fibril, and then we've gone to an off-image area, that is no body image, no blood image, and there are particles in there. Now, I could go back to that body image and hunt the tape, and I might find a couple of particles, because basically there are no tape samples that we have that don't have any particles on them. But there's no statistical number of particles on the body image. The body image is not caused by particles. It is caused what we see is caused by these yellow fibrils. Now, the image density changes a little bit from one point to the other, which appears to correlate with an expected cloth body distance, as you'll hear more about. But let me just tell you why the image is a little darker in one area than the other. It's darker not because the fibers are yellower in one area or another, but simply because there appear to be more yellow fibrils in the darker area, and the coloration of each of the fibrils is about the same darkness as all other yellow fibrils in the body image area. So we don't get a darker image because the fibers are darker yellow. It's only because there are more fibers in the darker area than there are in the lighter areas on the body image. Incidentally, here again, this is another body image with a slightly different backlighting. You can still tell that that fibril is yellow. And again, there's no particles in that region. As Al will describe to you, we know what the chemistry of these yellow fibrils are, and it's similar to the chemistry of just old decomposed linen. So what is there is a kind of a accelerated aging or an accelerated decomposition of the linen fibrils itself by some catalyst. When I say that there had to have been some catalyst there, the fact is that the whole shroud is aged, but the body image is aged more than the shroud image, than the background image, the shroud itself. So something had to cause that sort of accelerated aging, which caused those linen fibrils to be a darker yellow than the background. There doesn't appear to be any kind of indication that the fibrils are yellow due to a high temperature type of contact, because there are certain decomposition products which are prevalent with cellulose at higher temperatures, and by this I mean above about a 200 degrees Celsius, that do not appear on these fibers. We do find that stuff on the shroud, but in those areas where the scorches were caused by the 1532 fire. So we don't see any evidence of high temperature decomposition in the body only area except for where it intersects the scorch. This used to be kind of baffling, because if you think about what causes linen to yellow more rapidly, you always think of sort of scorching it in some way. But the fact of the matter is, turns out that once the chemistry was identified, I was showing you a picture here of Sam Pellicore, who insisted on doing all kinds of weird experiments. And he turned out that he could produce this same chemistry with, I don't know what the last count was, hundreds of things. And one of the things is just body sweat. A number of things start the decomposition off on a different slope than the normal one. And we've been able to reproduce this in a number of ways, but for one thing, most of these ways require time as an integral part of the process. In other words, initially there's no image when you put this thing on, and then after a while you start noticing an image with time developing on the cloth, because the cloth itself is aging, and this is just aging much more rapidly. And we've been able to accelerate the time base by raising the overall temperature of the cloth. Again, below that point where you start getting decomposition, high temperature decomposition products, but by raising the temperature above the normal ambient temperature, you accelerate all the aging processes, and we can actually develop images then. Now, one of the easiest ways to make an image is to use lemon juice, which is a weak acid. You've probably done this when you were a kid. You take lemon juice and you write a message on a piece of paper, which is cellulose, and then if you want the message to come out, you don't see it after you've written it, then you want to read it, you put it over a light bulb, you warm the cellulose up, and sure enough, this image develops. Well, the lemon juice has modified the cellulose. It begins to age more rapidly. That message would eventually develop in time had you not heated it, but what you've done is you've accelerated the aging of the cellulose so that you can read the image. Let me just show you a sample that Sam made by taking an aerosol, spraying lemon juice very lightly over his hand, and placing the cloth lightly on his hand. This is what he got. And I want to point out a number of the things that are on the shroud that are common to this sample that Sam made. For one thing, you see this characteristic striping of the image on the fiber itself. Secondly, the image is only on the topmost fibrils of the thread, and I forgot to mention that earlier, but the shroud image only resides on the top fibrils of the thread itself, and here we've been able to do that by just a light aerosol on a hand and lightly putting a cloth over the hand and removing it, and we get, and as I said, lemon juice is not the only thing, body sweat will do the same thing, and as will thousands of other things, I suspect, but at least hundreds of other things. And you see the placement of the image on the cloth microscopically exactly like it appears on the shroud. Indeed, the chemistry is the same. The other thing that I failed to mention when I was talking about that is that while we believe that there was some catalyst on there responsible for the accelerated decomposition, I say catalyst, it could be any sort of kinds of catalyst. Temperature is a catalyst, for example, but any sorts of catalyst. We could not identify any catalyst on there. We couldn't find anything but those things you'd expect of old linen. So if there was a catalyst, a chemical catalyst on the cloth at one time, it is not there anymore, or at least it's not there in sufficient quantity to be detected. But an awful lot of these kinds of things that will cause the linen to decompose don't stick around. So there's no reason to suppose that many of these things would be there anymore. This is a edge of a water stain. It looks quite a bit different than the blood images. For one thing, you can see that it's kind of coated the entire fibers there. It really penetrates the whole cloth. You can see, for example, here in the reversed image, there's one up there by the large blood wound in the side. And I just want to show you this one because it kind of dramatically shows that there are many, many examples of blood images on the shroud which are much darker in reflected light than the water stains. Now, I also show you this color separation. This was again made with those photographs that we made through different filters, and we've artificially stretched the color, but you notice that the water stains have a different color than the blood images. Right, what you're mostly seeing in this water stain region is the coloration of the cloth due to the presence of iron oxide. There's also calcium in there, but what you see in the way of coloring is mainly due to the iron oxide. And as Al will tell you, this used to be a way of making khaki. So that iron oxide is there due to the hard water that was used to extinguish the fire or whatever in 1532. We know these marks are associated with that fire. Presumably there was some water used to attempt to put out the box. At least that's what the comments say, and some of it got on the cloth. It was hard water, migrated to the edges, may have taken any sort of iron and stuff that might have been on the cloth already to the edges, and then it turned iron oxide and some calcium there too. Again, notice that it's quite different in its reaction to UV fluorescence. Here you see that the blood absorbs UV like mad, the blood image area, and the iron oxide doesn't do much. I show you this, but it's really just an example of many examples, and you can also read it in one of our first published papers on the X-rays. Interesting thing. Every single watermark showed up on the X-rays, every single one, but not one single blood mark showed up. Now here is a transmission photograph, again, of the shroud area, and you'll see that not only are the blood images darker in reflected light, but they are much more dense than the water stain areas. Now, if you don't want to believe me that the blood is made out of blood, let me just ask you this question. If the blood was made of a very thick paint, very dense like this, and was made out of mercuric sulfide and iron oxide, wouldn't you expect that stuff to show up on the X-rays, which are, we know, at least sensitive enough to pick up the water stains? You don't have to believe our chemistry to question that kind of a statement. Let me talk a little bit about the placement of the image. This is not based totally on the collected data in '78, but it's compatible with all that data, and we've done better things with it. But I want to talk about the density pattern of the image on the shroud. For one thing, it's been known since the turn of the century that the image, if you reverse it photographically, you get this sort of negative quality to the shroud image. Now, I will admit that I personally think that the negative image turns out to be a rather simple consequence of all kinds of processes that you can make images with. I've seen some very good mock-ups of the shroud, or ways of making a shroud by putting different things on corpses and statues and placing a cloth over it in different ways and producing an image which by its very nature has this negative quality. There's a collection of papers that comes out in a journal put out by the Center of Syndonology in Turin called Syndone, which I think means burial cloth or something like that. And I have been reading articles from that particular magazine for years and years, and I've seen some very fine negative images made by one process or another. All of those negative images, however, fail in both chemistry and the way in which the body image is placed on the cloth. Remember, it's quite superficial, and every one of those that I'm familiar with had something soaking through so that you could see the image on the back. But there are many, many techniques that have reproduced the negative quality of the image quite well. But let's talk a little bit more about the density pattern. Here is a picture of the shroud, which is a little bit truer to the kind of subtle way in which the shroud image is placed on the cloth. It's very light. This, in fact, was photographed through a blue filter and color corrected in the print, and so it is enhanced. And yet you can see just how subtle it is. Now, some of the black and white pictures that we have are even more subtle, and this subtlety is necessary to draw out, try to get as much information in the shades of gray as possible. And this is one of those which is not as aesthetically pleasing as you might have seen on things like, like the exaggerated images. It carries more information, but yet it has the same quality as a number of those images that we've looked at. But it turns out that the image appears to be darker in regions that if a cloth was over a body, you expect it to be closer to the body, and lighter in those areas where you'd expect it to be further away. And you'll hear an awful lot about this tomorrow in a paper by John Jackson, where he'll go into excruciating detail, more detail than you ever wanted to hear about this particular topic. But let me just say that when we look at density patterns on the image, compare it to expected cloth body distance from subjects wrapped in cloths and placed like would be indicated by the position of the image on the cloth. And incidentally, let me say that I have been directly involved in wrapping a whole lot of people, and it's incredible how easy, when you get the right size person, it is to simply lay that cloth down and have the features that are depicted on that cloth lie right over the correct anatomical location on the body. And when I read these articles that just keep being repeated about how distorted the image is and how distorted a human body would have to be to fit that, I can only say that those people have not done these experiments, because you get the right size body in there, a normal human body, not a distorted person, and those things just match right up. In fact, it's uncanny. Well, when we do this comparison, then plot it, we find kind of a comparison between expected cloth body distance and the darkness of the image. Yes, there's some noise and scatter on that image, but that relationship is so universal over the body that we've been able to graphically demonstrate it on a VP8 image analyzer by plotting the density as vertical relief, and you can see that it looks like a body image. Now, we feel that that is compatible with the notion that the shroud wrapped the body. Okay, so we make that. We're not saying it's proof, we're saying it's compatible with that notion. Furthermore, when we make an image like that, it's kind of flat. I've sort of depicted here where we go from the density pattern through the correction curve down to the image that you've just seen. And then if we fold in, that's a nice accidental use of words, but when we add then the distortion due to a normal drape or an expected drape of a body over, cloth over body, we get an image that looks a lot more human-like. And this is an example of that. It's not a very good picture, but when we did that, added this distortion, kind of distortions that you would kind of expect from a cloth wrapped over body, to that density pattern, we get a fully three-dimensional image that looks like a man. And furthermore, when we look at what we had to add to that to make it look like a man, it looks like a cloth wrapped over a body. And if you don't believe that a cloth can be wrapped over a body like that, let me show you something. This is a cloth made by Kate Edgerton to the specifications that the weave and the density and so on of the shroud. And this is laid over a statue there, and you can see that the cloth is fully capable of distorting into the way that we had to distort the image in order to get it to look more fully three-dimensional. So we think that there's not only the notion of cloth body distance is compatible with the density images, but also the notion that the cloth was wrapped over a fully three-dimensional body, because that subtle distortion of the cloth that had to be added to get that is compatible with the way a cloth normally folds over a body. Okay, that's the last of my slides, and now I've got some closing comments. This is basically what we found. I think that what you've seen, what I've shown you, and what I tend to believe is that a lot of the things that we find are most simply explained by saying that the shroud is wrapped over a body. Or if you think of it another way, if I take the notion that the cloth was wrapped over a body that was beaten up, I would get these kinds of blood images on the cloth, transferred to the cloth by contact with the blood. We see blood on the cloth. The notion that the cloth was wrapped over a body seems to be consistent with the density pattern. There are all kinds of natural body exudations that can cause the coloration of the image that we see on the cloth. Does not mean that we know how the image was formed, because there are problems. You'll hear about these problems. But the point is that I don't think it's a wild speculation to say that the notion that the cloth wrapped a badly beaten body is a unreasonable compatibility match. It's not proof, no, but it's reasonable. Now, let me also mention something that I'm sure Al mentioned, and that is that when we remove the blood from the shroud fibrils, there is no body image under the blood. Now, this is apparent also in some photographs where there is blood serum away from the dark blood area, and you can see a halo around that. And that little halo is the best preserved fibrils on the shroud. They are less aged than the off-image control area because they've been coated and sort of preserved, and the normal decomposition has been delayed. Again, you'll hear about that. So here we have this body image. There's no body image under the blood marks, and it seems to me the blood went on and it prevented the body image from transferring. So all of these things are consistent with the notion that there was a body wrapped under the cloth, but it doesn't say anything about whose body was there or anything else. Now, maybe you think that, well, if we've had these findings, why is there so much controversy? How come you haven't made these things public? Well, let me tell you how we have made these things public, and I think we've done it in the only responsible way. For one thing, we cooperated with the National Geographic, and in June of 1980 there was a very fine article there that contained all sorts of things, including photomicrographs, and clearly demonstrated, for example, that the body image was yellow and not red, and yet people are still laboring under the impression that it's red and so on. But let me tell you what else we've done. For one thing, we have attended technical meetings. I've mentioned already the annual meeting of the American Chemical Society that Al and I presented a paper at in front of chemists, and some fairly hostile chemists initially, that in the end thought that we had something to say. Very well received. We've also taken our image analysis work and presented it at an IEEE conference, which means something, the Institute of Electrical and Electronic Engineers, and that's the people that do image analysis. We presented our findings there. We have, in fact, spoken at the Canadian Forensic Society, which does things like look at medical forensics and things like that. I personally have spoken at many American Chemical Society meetings around the country. Al is on the American Chemical Society guest lecture tour provided by the national headquarters. I've spoken to Sigma Xi, which is the Research Society of America that is devoted to scientific research, many times. I never refuse an invitation to speak at a university or a technical society. I do refuse invitations to speak at religious groups. I don't speak at religious groups. We've done everything to get the word out that we can do. We're not very good at writing popular books, but in terms of writing down what we have said, let me just give you a quick rundown of our technical papers. And when I say technical papers, I mean that many of these are in scientific refereed journals. And if you're not familiar with the process of getting a paper in a refereed journal, then I can only say to you that you are not familiar with scientific research. It is a difficult process in many cases, especially with controversial issues, and we have subjected ourselves to that kind of test, and we've been successful in publishing our work in technical refereed journals. Incidentally, many of you will probably be surprised to learn that a scientist pays page charges to have those articles printed. We don't get paid to do that. Now, let me just start out sort of historical and say that in 1980 there was a paper published in X-ray spectrometry, which is a journal of X-ray fluorescence type of work, on the X-ray fluorescence work that we did. Also in 1980, there were a series of articles in Applied Optics, one of them explaining the purpose of the test and overviewing what we did, then a test, and then a paper talking about the infrared reflectance and fluorescence work that we did, a paper on the ultraviolet and visible reflectance and fluorescence spectroscopy, and a paper then a month or so later in Applied Optics again on the blood, entitled Blood on the Shroud of Turin, talking about the preliminary work that we had done on the blood. There was an article in Archaeology talking about the photomicrographs and a very nice color layout of what was found through the microscope. That's not exactly a scientific journal, but it is read by people in that field, and I think it was a nice format for giving color pictures through the microscope, and also that article was reviewed. There was an article in Industrial Research and Development talking about the instruments that were used to study the shroud. In a journal called the Biological Photography, published in England, the Journal of Biological Photography, there was a whole series of photographs in the ultraviolet fluorescence and just normal photographs that were made on the shroud. In the Canadian Society of Forensic Science, which is one of the foremost forensic science journals, was a paper on the chemical investigation of the Shroud of Turin. Then there was a series of articles that appeared in the proceedings of an IEEE meeting where we presented a lot of our work. There was a summary of the results, talking about the test points on the shroud and where we did various things, talking about the scientific photography that was done, also talking about image analysis, 2 papers in that area, and then a paper on talking about these distortions that we found in the image. And then in the Archaeological Chemistry 3, which is a book that comes out every once in a while in archaeological chemistry, put out by the American Chemical Society, there was an overview article called A Comprehensive Examination of the Various Stains and Images on the Shroud of Turin. And then in Applied Optics, just I think a year or so ago, there was a paper detailing the work on the image distortions and density patterns and the, quote, three-dimensional nature of the image and its compatibility with the notion of wrapping a body with a cloth. Now, what I've told you are 20 scientific papers. Quite frankly, I don't know what else we could have done. And let me also just read from the comprehensive examination a kind of a closing statement that says, in the end, the question of authenticity of the shroud as the burial cloth of Jesus of Nazareth remains open-ended. We should all keep in mind that science is really not in a position to ever prove categorically that the shroud is authentic, i.e. that Jesus' burial cloth. We have, however, examined the probability that the shroud was artistically produced and examined and find it improbable. The question is left so that those who wish to believe it's authentic are not hindered by scientific objections to doing so. However, without proof of authenticity, those who choose to believe that the shroud is not authentic are also free to do so without scientific objection, provided they do not assert a production mechanism that is excluded by the information now available. And I think that pretty much states it. All we have done is provided a discriminating set of data. That's all we've done. We're not trying to grind any axes. That's all we have done. Now, where do we go in the future? Well, obviously, since the thing doesn't come up in obvious forgery, we need to get a carbon date. It'd be nice to collect some more sample. People have some questions about what we did. Let's get a whole lot more sample. Let's get a whole lot more people involved and see if our findings on the chemistry are right or wrong. Now, that's all we have done. And if I close with what I started with, I personally cannot understand why in the media our credibility is trying to be attacked. The only thing I can think of is that those people who are trying to cast doubts on our credibility and trying to slander our work by slandering us are doing so because they find something objectionable in the data. And it probably because it contradicts what they believe was the cause of the image. Otherwise, why don't they attack the data and not the credibility of the people? We've done everything we can to responsibly, in the scientific framework, tell what we've done and to report our results. We have produced a discriminating set of data against which various hypotheses for the formation of the image can be tested for compatibility. That is all we have done. Thank you.